Okay, so very good morning. It's just gone 9:30 a.m. here in the UK. My name's Kim. Can you just pop in the chat? You've got a little chat function. Can you just say hello, hi in the chat just to let me know that you can hear me okay? Great, thank you. Somebody said hi. So, hello, good morning, good morning, good morning. Whilst we're waiting for people to log in, we'll wait for a few moments before we start the actual lecture. If you're happy to, could you pop in the chat and let us know where you're joining us from? It's really interesting for us to know where our audience is in the world. This is the first of our virtual taster lectures of this series. Somebody's joining from Morocco, Woking, Hampshire, and Hong Kong. Wow, that's a fantastic spread. Thailand. This is really exciting. So we've got and Singapore, Thailand, Hong Kong. So that's very interesting to see that we've got people from that part of the world. A very, very warm welcome. I hope your week is starting well. That's very helpful for us. It means we can get a bit of an idea of where people are joining at this time in the day. The rest of these sessions are scheduled to happen in the afternoon, at lunchtime. So we always try to do this to get a rough idea of where people are coming from. So now that we're sort of mainly we're logged in, I'm going to launch a little poll just to see. Just bear with me. So polls and quizzes. This is just to see. If I launch this is just going to ask you, do you have an offer to study? What sort of level would you be? Would you be more interested in a degree or a postgraduate, like an MSc? That way we can get an idea of whether people are coming to us as a degree student or maybe an A-level or an IBAC student. It's really helpful for us to know that. And then the other question is, do you have an offer? So this has been promoted widely, this webinar, so it's interesting to know whether people are offer holders with us or elsewhere at UCL or not at all, prospective students. So I can see that people have filled that in, and mainly it looks like people are intending to study a degree, and the majority of people don't have an offer, but I can see that people have an offer. And also, we just threw a question there to ask you if you know what an enzyme is. Again, just trying to find out, just roughly for our interest. But thank you very much for filling that in. That's really, really helpful. So what I'm going to do now is end that poll, and then I'm going to start talking about this session a bit more. So as I said, my name's Kim. I'm the Outreach and Communications Coordinator here at UCL Biochemical Engineering. So as you've already discovered that you've got various functions. If you want to reconfigure the screen, you can do that. You can have myself and Ivana speaking or the main screen. If you look at the bottom of your device, whether you're on a tablet or a mobile, you've also got things like Q&A and chat. Some people very kindly use the chat already. You can put your questions at any point in this session, but please use the Q&A function. It really helps us to keep monitor the questions that are coming in. We can sometimes see similar ones that are gathered together, and I might be able to deal with some that directly if necessary. Please put your question in the Q&A. You can put them at any point in the session, but we'll come to them after Ivana's given her talk. If you have any technical problems, if you can't hear me or there's any kind of an issue with the screen or the slides, please pop in the chat and let us know, and myself and Ivana will see that. So what I'm going to do now is just hand over to Ivana. Oh, and also there's one point I want to make as well, which is really important. If we cover anything in this session which you feel you don't know about, so anything scientifically or engineering wise, if you'd come and study a degree with us, we will take you to this level. So sometimes people attending these are saying, I don't understand this. If there are any parts of this you don't, if you don't understand, if you don't get a term or something, please let us know. But generally, when you study a degree at UCL Biochemical Engineering, what we do is we take you to the level where you're understanding these things. So now that I've gone through all our housekeeping, so to speak, I'm going to hand over to Ivana, who is going to give the presentation. I might turn my camera off, but I'll be here in the background, okay?
Good morning, everybody. Welcome to the session. I'm Ivana, and today I'm gonna, present you the first virtual lecture. Let me just share my screen. Okay, I guess you can all see my screen. All right, so I'm Ivana, and I'm a research fellow at the Biochemical Engineering Department, and this is the first session of a series of virtual taster lecture. They're gonna really show you what you can do about chemical engineering, what we are doing as researchers in this department. So today the topic is going to be high throughput screening, high throughput process development, and how myself and my colleagues are using these to build better vaccines and quicker. So before going into more details with the presentations, I wanted to show you something about my career path. You may, perhaps someone of you is already an undergrad. You may have a slightly different background, but are interested in biochemical engineering. I think it's important to show you how you can have a different background, but then end up in this department or vice versa. So I'm actually not an engineering, I'm a pharmacist. I got my degree back in Italy in 2017. During my degree, I did add some experience as a community pharmacist, but I also did study for 6 months in Paris. There I had a research experience. I actually worked in a lab on nanomedicine. It was actually that experience that really helped me understanding that I wanted to pursue a career in research and development. So after my degree and after an experience as a pharmacist, I got my scholarship at King's College London. So I moved to the UK and to London in 2018, and I started my PhD in pharmaceutical science. My PhD was part of a cohort of a doctoral program called LIDO. And during my time at King's, I did work on nanomedicine for lung delivery and lung therapy. So after my PhD that I got in 2023 and then moved to Pfizer, and I worked there as a senior scientist. And then later in 2024, I joined UCL in a joint biochemical engineering department as a research fellow in the VAX Hub, which is a hub, a UK-led hub made of different universities. There's lots of postdocs, lots of researchers. Everyone is working on a big project, which is really about vaccine development and how to speed up vaccine development. So let's get started. Kim, if you can just go ahead with the first questions. Before getting started, I want to make this presentation engaging. I want to get some feedback from you. So the first question that is gonna come up, I think it's coming up now. Kim?
Yep, it should be with you. In fact, some people are already filling it in.
Oh, okay. I think I can really say that. Let me perhaps just, okay, can you perhaps, Kim, just tell me about the answer for from the pool for the first question, because I can actually see
Absolutely.
the
So the question is, how long do you think it takes to develop a vaccine? So most people say it takes more than 10 years, roughly 44%, and then it's an even split between 2 years and 6 months. So fairly
amazing.
similar thoughts on
Okay.
that. Okay.
We do have some good answers here, and I think you'll kind of like get some information throughout the presentation, and will allow perhaps you change your answer, but let's see. Let's go to the second question, Kim. Thank you. Which was, how much do you think you know from 1 to 10 about vaccine development? And I think whatever number you're actually putting into this answer, I do hope that we're going to increase that number even by 1 unit by the end of the presentation. What kind of numbers are coming up, Kim?
Well, it's a fair spread. I'd say most are sort of like 3 to 5. People think they know a fair bit about it, but nobody thinks they know an awful lot about it. And then there are a few people who say they don't know anything at all. So at the moment, it's sort of like a bit like that.
I'm glad. So I do hope you're gonna get some information from this lecture, and you're gonna hear more about what we are doing about chemical engineering. So let's get started. First big question is why vaccines matter. So I'm sure you all got some vaccine shots when you were kids, perhaps even like more recently. But I think all of us really have a clear idea of why vaccines matter because of one of the latest events really. A few years ago, I'm sure you all remember how COVID-19 pandemic really hit our lives and changed our lives. And I'm sure you all remember what was the situation before the vaccine was approved and after the vaccine was approved. So I'm sure we all agree on the fact that vaccines are a very effective public health intervention. They can eradicate or control diseases such as smallpox, polio, or for example, COVID-19 as well. And of course, being preventive treatment as well, they can reduce healthcare costs and economic burden. So the challenge, why vaccines take so long? Some of you actually answer about for the answer about how long does it take, they answer 10 plus years. And that actually was the correct questions. Overall, usually takes so much like time, but why? So this is the vaccine life cycle. It's an overview of all the different phases that usually researchers need to go through and pharma companies to develop a vaccine. So we start with the basic research, with the discovery and the preclinical studies, which are studies in cells and in animals. And then once there's data about the safety and efficacy in cells and animals, the second big phase is the clinical trial studies. So the testing on humans, volunteers or not volunteers, like people with diseases. Once all the informations are collected, and if those are good information, the following step is the approval. So collecting all the documents for the health authorities, and then the final phase is the phase 4, which is the post-approval monitoring. So you can see how long is really the process, and of course bear in mind that each of these steps has got additional steps. So the overall vaccine life cycle is very long, and the question is, what can we do to make these quicker, to kind of like reduce really the time of development? So CEPI, which is the Coalition for Epidemic Preparedness Initiative, has actually started already doing these and trying to reduce and cut the development time. It's called 100 Days Mission, and the aim is advancing vaccines for both known and emerging viral threats, strengthening the platform technology. So the idea is to develop a vaccine platform that can be flexible, that can be adaptable based on the disease and the threat is actually emerging. And that's something that myself and my colleagues at UCL are actually working as well. The idea and the researchers at VaxUp is to work on a platform, so on a type of vaccine that can be adapted whatever threat will come up at some point in the future. So I'm sure most of you, if not all of you, knows that a vaccine is made of antigen. So it's a part of a pathogen that will induce an immune response from our body. But in the reality, a vaccine is not only an antigen, but there are so many more other aspects that researchers work on. So first aspect is the formulation. So formulation is really what makes a product can be delivered to humans. So only an antigen can be delivered to humans in a vaccine. We need excipients, we need adjuvants, we need a whole product. And of course, the characteristics of this product will impact the route of administration. Are we going to administer this vaccine via subcutaneously, intramuscularly, or by inhalation? Are we going to have just like a suspension or perhaps some nanoparticles or microparticles in the vaccine? Is the vaccine going to be a solution or lyophilized, so like a powder, and then the GP or the nurse is going to actually add some, for example, sodium chloride to the powder? So there are so many different aspects that will impact the dose as well. So how the vaccine is bioavailable? Do we need 1 shot, 2 shots, or like what's the interval between doses? All these aspects, so formulation and dose, will also impact the stability. We need to make sure that the vaccine will be stable on the shelf of the pharmacy or the GP office for a long time. Another key aspect, which is critical actually for researchers about chemical engineering, is the scalability. So I hope you can see here. Actually, let me do like that. Okay, so all experiments, they start in the lab, right? We work with small volumes usually. In this case, we do have a shake flask, and there's about 100 to 100 milliliter of volume in this shake flask. But of course, when we think about producing vaccines for population, for like many people, you need to increase this volume, and that's how we call scalability. So we need to go from small volume to bigger and bigger volumes, and this takes lots of research really, which is actually undertaken at biochemical engineering as well. Another aspect that researchers work on in this department is the downstream. So once for example, bacteria that you have engineered is actually able to produce the vaccine, you need to separate the bacteria and the product and make this product pure. So of course, that can actually be administered to patients. And finally, there's what we call cold chain. So cold chain is what we refer to as the fact that vaccines very likely need to be stored at -20 or -80 from the time that they leave the manufacturing site until the time they actually administer. So in all these steps, the transport, think about if a vaccine is manufactured in, for example, in Europe or in US, and then has to be transferred to Asia or to Africa. Each step of this transport needs to be kept at, for example, -80. This is something very difficult. And lots of researchers are actually trying to make ways of ensuring that the temperature can be, for example, room temperature or slightly higher instead of -80, which of course is very difficult. So you do see how many really pieces of research are required to make a vaccine actual product that can be developed, that can be delivered to all of you. So the traditional approach, how can we move from the traditional approach to something that is quicker and is more efficient? So the traditional approach is one experiment at a time. So I'm currently working with bacterial vaccines. So this means that I use a bacterial coli, Escherichia coli, which has been engineered, that contains plasmids inside, and this bacteria is able to produce a vaccine. But of course, these bacteria need a specific condition. It needs a specific temperature, a specific media, or a specific shake. So all these conditions needs to be optimized, and each condition will be tested in a single experiment in the traditional approach. Of course, you do see how this is a very, very lengthy process. It will take just so many months. So the question is, how can we make this quicker, just to increase really to speed up the vaccine development? So I'm currently working with my colleague, biochemical engineer, which is called Ryan Meller, and we are working with an automated system using a Tecan Freedom Everywhere. So here is a video where you can actually see how we're using this system to just make all experiments just quicker, because the alternative would be myself, for example, doing all these steps one by one, which I do assure you that it would take just so much longer. So this robot, what is doing is taking tips at the moment, is withdrawing volume from a reservoir, is dispensing volume in a deep well plate, and then discarding the tips, putting the lid on the plate, and then moving the plate onto a bioshake, which you do see at the back, which is gonna shake and warm up the plate. Then again, dispensing volume to a different plate, which is a smaller one, and then transferring this plate into a plate reader, measuring the absorbance, and then putting the plate into the centrifuge and spinning down our samples. So, of course, this was a slightly more accelerated video, but you clearly can see the potential of this platform and how just would make things so much quicker. So, what we're going to talk about now is the high throughput process development automation. So, of course, you did see some bit of the automation, but what does it mean also high throughput process development? So high throughput process development is a systematic approach. It does use laboratory automation, what we just seen, the Tecan, but also miniaturization. So if you do see a couple of slides earlier, I did show you the shake flask, which is a way of testing, for example, media for bacteria that contains about 100, 200 milliliters. It's a quite big volume. This means corresponds to high time, lots of money, consumables, and so on. So the strategies really is like change the volume and do everything smaller. So some consumables that we currently using, for example, are 96 well plate or 384 well plate or deep well plate, which can allocate a bigger volume. So the idea is to combine the automation, the miniaturization, and the parallel experimentation to really increase and speed up the vaccine development. So the key advantages of high throughput process development are speed, of course. We do really reduce massively the time for process development and optimization. Efficiency, we can screen so many parameters at the same time that otherwise would be just like impossible really with traditional methods. And then quality by design. So really provide extensive data that help in establishing very robust experiment. So what I'm currently working on is media optimization studies. So as I mentioned, I work with a bacteria which is called Escherichia coli that is able to produce our vaccine. Right, bacteria needs a specific media. They needs to be like a feeding really. And these media can have a fixed composition, so you can just buy really from suppliers. But from that media, you don't really know what's the components. So the idea is to have a really detailed list of ingredients of these media, specifically the composition, the concentration. You can adjust this concentration, this composition based on your needs. So For example, what I'm currently working on is improving the media and making like GMP compatible. So GMP stands for Good Manufacturing Practice. And it's important that if we think ahead, if we think about the approval process, the regulators or the health authorities, which can be, for example, the FDA or the EMA, they will want the media that you use to cultivate your bacteria to produce your vaccine that non-containing any animal-derived components. So there's a really, really big study that needs to be done to make sure that the media is the best you can get and is really allow the bacteria to be just really happy and to produce as much vaccine as possible. So now there's another question for you. So this is a plate, and it's a plate I'm currently using for my experiment. So the question, if Kim, you can pop the next question, is how many conditions, how many experiments do you think I can actually test using this plate in my experiment?
That's just been launched.
Amazing, thank you.
Just want to see the picture of the plate isn't on anymore. There we go.
Right.
Okay, well, yeah. Okay,
Can't see the answers, but yeah.
so the vast majority of people, like two-thirds have said 96 conditions. About a third have said 28, and then some people have said 8.
Amazing. Okay. I think those are great answers, and I'm going to
Thank
just
you for
tell a bit
filling that in,
more
by the way.
about.
Thank you.
Sorry?
So I just want to say thank you, everybody, for filling these in. It's really helpful.
Oh,
It
yeah,
really
of course.
does. Cheers.
I'm sorry, I can actually see the questions, but thank you, Kim, for launching that. And yeah, I mean, most of you actually gave us the right answer. So using this plate, which is a 96 well plate, I can actually test 96 different conditions. So each of these, you can see, I mean, colors are different here. Each of these well, which can contain about 100 microliter, is a different experiment. So you do see the potential of these high throughput process development, whether if I had done these experiments with the old traditional way, I would have done perhaps at least, I don't know, 96 different experiments, whether in this case, I can use one plate, I can do the experiment only once, but each of these well is going to give me a specific answer. So here is an example, for example, well A12 and well G6. So if you do look at the underlying factors, which are the ones that I'm actually looking at, so changing, those are the variables, right? So if you do see just for these wells, you do have a different glucose, sodium chloride, peptone, and yeast in these two wells, and these concentration will actually be different for these factors in each of these wells. So this really was to show you the potential of high throughput process development and how it can really help us reduce cost, as well as, for example, with the miniaturization. So high throughput and miniaturization in my research really go hand by hand. So I hope you did get the potential of this technology and how these can really help speed up the process development of vaccine, which is what I'm working on at the moment. But of course, I think it's important to highlight that high throughput tools are actually being used in so many other aspects of vaccine and drug development, drug discovery, process development, what I talked to you today about, but as well as explore the drug design space. And overall, they do have an impact in reducing the product development timelines. So I hope you enjoyed the presentation, but just to conclude, I want to really show you just briefly what all my colleagues at Biochemical Engineering are actually working on. And I think it's important if you want to join the department and if you to join undergrad or postgrad or perhaps at some point also a PhD, I think it's important for you to know that lots of researchers are in the department. They're all working on different topics such as mammalian high throughput virus production, or there's another research group working on bioprocessing and scale up for cultivating meat. You're gonna actually, there's another taster lecture coming up in the next week about this from my colleague Ben, or as well bioprocessing and scale up for CAR T cell on, of course, what I've just talked to you today about, which is the vaccine development at the microscale level with the automation and the scale up as well. So I hope you enjoyed the presentation. Please feel free to reach out if you have any questions or like connect with me, or if you want to hear more about the VaxUp and perhaps at some point also join the VaxUp. Here is the QR code as well. So yeah, I do hope you enjoy the presentation, and we're gonna go through some questions if you have any with the Q&A session. Thank you.
Fantastic. Thank you, Ivana. That was brilliant. I really enjoyed that. I'm very lucky I get to sit in all these webinars, and I've learned so much through these. It's really, really great. Okay, so we've set aside half an hour to take any questions that you have. If you've answered all your questions, then we won't keep going. But we're here for anything you want to know about Ivana's work, her vaccine, and presentation. Please use the Q&A function. As I said, you've got a little button down there so you can ask questions. Also, thank you, Ivana, for talking about the other webinars that we've got coming up in this series. What I'm going to do, I think if you've signed up to this, you would have seen a link to the other ones as well. But this is being recorded. All the sessions are being recorded. So if you sign up to any of them, I'll send a link out afterwards so you can watch them back. They will get uploaded onto YouTube, and I'll create a single page with all of the presentations on it so you can watch them back there. I'll also send a link out to everyone registered so they can watch it back on Zoom, because I know not everybody in the world can access YouTube. But I think everyone can watch back on Zoom. If that doesn't work, we'll find another solution for you. But we are keen to make these available for everybody to be able to watch. So we've had a question come in, which is a really good one. And it's something which a lot of prospective students ask generally about our laboratories. But I'm going to hand that one right over to you, Ivana. This is a really good question.
Yeah, thank you for the question. So the question is, what does a typical day in your job look like, and do you enjoy it? Well, first of all, it says also, thank you for your talk. I hope you enjoy it. So I've been a researcher for a long period of time. As I mentioned, I did have research experience previous to my degree, and then it's 4 years of PhD, and then my experience as a researcher at Pfizer, and then now has been about almost 2 years at UCL. So at the moment, of course, because of the previous research experience I had, my day looks a bit different. I was in the past, but at the moment, what I would normally do is that so I do have, let's say on a Monday, I would go to the office. Very likely, I had a list of things that I had to do that I wrote down on Friday, just to make sure I start the week properly. So I would just have a look what experiments I did plan, go to the lab, start preparing, for example, some buffer or some assays, set up some experiments. Have some meetings with my colleague that I mentioned, Ryan. I really work closely with him. Some meetings with my manager, some other activities that are linked to the VaxUp. That can be, for example, engagement activities. And then the day after, I would perhaps do other experiments. There's usually at least a day or a couple of days that I would just be in my office or at home with my computer, just looking at data, because you just can't do experiments every day. At some point, you have just to sit and see what you're actually getting, what kind of results to really decide, like some informed decision. Otherwise, that's just, you need some kind of direction. But yeah, I think it's quite varied. Of course, the lab is a big part, because I do research, and my research is a wet lab research. So definitely I need to be in the lab, but not every day. But yeah, I definitely really enjoy it, and I think it's a good career path. So yeah, I hope you'll choose it as well. So thank you for the question.
I just want to jump in there and say, Ivana, you're mainly working in the pilot plant. Is that right? That's where your work is mainly focused?
I do work, yes, in the pilot plant, but also in another lab where the Tican, which is the robot that I show, is actually there. So because I do different kind of I do have different, I work on different research areas. So definitely vaccine development, but I do work on the micro scale, which is what really I show you today. But I also work on a scale up, which I didn't talk to you today about, but that is bigger volume. So depending on the experiments, I can be based in different labs. We do have lots of labs in the department. So I do move around quite a lot in the department. But yeah.
Fantastic. And just for prospective students as well, one of the things that we really try to do is to give students a real experience of working lab. So our pilot plant is actually a scaled down industrial facility. So we have bioreactors and processes being able to purify and refine the product, and it's really to train people in how you work in a professional, full scale facility. So that's we're unique in the UK in that we have that in university. We also have other laboratories that focus more on something stem cells or synthetic biology. Some of the other talks that we have coming up, we'll have other people talking about their work in those labs particularly. So we hope to give you a real idea of what researchers are doing what in which places, but we really do try and give students a real experience of working in these laboratories. And Ivana has experience working in these laboratories and wants to carry on. Some students find that they just don't like working in the laboratory. They much prefer working with modeling or with design or consultation. So we really want to give students the chance to find out what they like doing so they can choose what they do like doing, what isn't for them as well. So we really provide those opportunities. It's really good fun. Also, we do Pilot Plant Week where you work as teams and you get a challenge and you have to report back on it. And that's usually really good fun. Very busy. Very interesting. So, okay, I'm going to
Yeah.
hand over to, there's another
Yeah,
question at the top here.
we do have quite a lot of other questions. So I'll go with the
Yep.
other one. So do you have any teams working on research into vaccine uptake and whether this high throughput process increases or decreases uptake? Or is the university's work focused specifically on the development of vaccines? So I'm not sure what you mean with uptake. I'm not sure if you mean kind of like how the vaccine works in the human body, or if uptake you mean kind of like looking at the uptake of vaccines by cells. I definitely say that there are some research groups that do preclinical development, being preclinical with like looking how vaccines interact with cells, which can be like mammalian cells, different type of cells. But yeah, in the department, there's, I think the only part of the preclinical studies that is actually taking place is interaction with cells, but no animal studies. The department doesn't have like animal studies involved. So I hope I did answer to your question. But yeah, so the other one is thank you for the presentation. Okay, could I please just know a little more about the testing you do? Do they usually involve clinical trials? And before that, how can you make sure that the risks are minimized in the lab before sending them off to the patients? Okay, we definitely don't do clinical trials in our department. Clinical trials involve, of course, very likely being a hospital or like a health research facility. You need to have there's like lots of regulation and regulation around the clinical trials. So that's something that you would probably see in like CRO, which is clinical research organizations, or pharma companies. So what we do at the department is more like in the preclinical phase, the downstream processing, the upstream processes, and of course, where they do clinical trials, they do lots, as I show you, one of the slides at the beginning of the vaccine life cycle. Vaccines and overall all drug products, they just really undergo so many tests and like assessment, and at the end, really a small amount of molecules or that where they started at the beginning, in that kind of like very long phase, they actually reach the market. So if you start from like 1,000 molecules, perhaps just two will reach the market. So the assessment in terms of risks is really detailed. So yeah, there's lots of risk, of course, in terms of like safety and risk assessment. I can't really go into much detail because that's not really my area of expertise, but I can assure you that there's lots that is going on. On the other side, that's something that is more linked to pharma companies or clinical research organizations. That's something that is not happening in the department.
Cool. Just actually, perhaps just clarify that one of the things that biochemical engineering is the discipline of taking a discovery. So by the time a vaccine or a drug or whatever it is, maybe it might be a protein or a plastic product, the challenge for biochemical engineers is finding a way to manufacture that and to make it in scale. And we do it with biology. So the work in like discovering a drug or a vaccine will have been done, the clinical trial will have been done somewhere else, maybe even at UCL. It happens a lot here. But then the challenge is how to make it in scale in time. If you can do that, then you can have a huge impact on global health. So I just want to jump in quickly and say there's somebody's asking about doing summer placements or PhD studentships. What I'm going to do is I'm going to pop my email address in the chat. In fact, it's there already. Get in touch with me directly if you want to find out more about PhD works, because we have a PhD admissions tutor who'll be happy to talk to you about that, and we have events for them. But we do summer placements. We do them through Interscience and Nuffield. And we're also, VAXUP has an outreach team as well. So we do it, but there is a process and we go through other organisations. So again, if you're interested in that, get in touch with me directly and I'll be happy to do it. And somebody's asked about PhD funding. We do have funded PhDs, but again, it's probably best if we discuss that offline. So just email me directly and I'll be happy to talk about that. But great that some of you are thinking about PhDs already. That's really good.
Amazing. Thank you, Kim, for taking those questions. Yeah, I do have two questions that perhaps I can answer them together because they both kind of like are asking about the differences between research, biochemical engineering, and pharmacy. One question. And then the other one was asking, is biochemical engineering more the later stage of development and how does it differ from a microbiologist's role? So I think both of them are really good questions. And I think before, like all my studies and my research experience, I was also struggling to really try to understand what you do and where, because I mean, the whole research that is around drugs and vaccines is really it's just like massive. And I think my experience in corporate really allowed me and helped me to really understand what's going on there's just like, of course, when you work in corporate, like in pharma, you do have a different perspective because you do see projects and drugs then going to the market. But I definitely say that there's perhaps when I was during my PhD, I was at the Institute of Pharmaceutical Science department. So lots of research that was actually taking place there was still lots of like around drugs and how they interact with cells, for example, but it was lots of like animal studies involved as well. So for example, really looking at how the drugs, so like a new modality, which is really what I worked on. So when I say new modality, we're actually referring to, for example, nanoparticles or microparticles or like liposomes. That's all that area where it's called new modalities. So really looking how these new modalities to interact with the body is actually the drug being able to be released over time, which is like control release, or like really reach the target. What's the bioavailability of the drug in the body? So I'd say that the pharmacy kind of like the pharmaceutical area, at least the research is actually taking place in like pharmacy department is more close to that. But on the other side, well, the skills that you can get with like research experience are really like transferable. You can just be in a department but not really do something that is strictly connected to that department. It really depends on the supervisor or the investigator you work with, because there are some people that can do different work. So I think it really depends on the department and on the supervisor you have. But definitely like what is actually taking place at biochemical engineering, I wouldn't say it's like later stage of development. So if you with later stage, we probably may refer to like clinical trials. Everything is at the biochemical department is still like really linked to preclinical development. But definitely like there's a lots of people working on scale up, which is something that usually in pharma companies would take place later in the development. So when you have a radio molecule, it works in like cells, it works in like animals, and then you decide how I'm going to manufacture this drug. So I think the department has a really tight link with manufacturing, which is really how we go from like small volumes, 100 milliliter to 2000 liter. That's something that is actually taking place in a department. We do have some equipment as well that allow research at these bigger volumes. So if you're interested in that kind of like aspect, more is really highly linked with manufacturing. That definitely is happening in the department as well. I hope I did answer to the question, perhaps a bit long answer, but yeah. So
Sorry, just
Let me just.
whilst Ivana reads that, just to let you know, we did have a question about degrees and entry requirements. I put the link to the page on our website, which has got our three. We've got three undergrad degree programs. There's the Biochemical Engineering BN Gem Eng, which is like a 3 or 4 year version of the same thing. And then there's a BSc Bioprocessing with New Medicines, Business and Management. They've got different entry requirements, different levels. So the best thing to do is have a look at the prospectus pages. You'll see full details of everything that's required. And you can see all the modules and programs as well. If you've got any more questions, I put
Yeah,
the
there's just a couple more, which I think we have the time to answer. So one is, what role does automation or technology play in evaluating the data from your experiments and high throughput process development? It's actually a good question. I'd say that, so I don't have personal experience. I do have a small experience, but not good enough in coding. But of course, all data that is produced, for example, with the TCAN, so with the robot that I show you, that is processed by actually my colleague, Ryan, which is really good at coding. So of course, if we think about that plate that at 96 conditions, right? And for example, sometimes we tested 96 different bacterial growth and for like 24 hours. So at the end, you have 96 times 24 hours like data. That's a lot. And of course, if you were to do this manually, it would just be impossible. So automation and technology really plays a role in this aspect. And all data is really processed very often by like using Python, as I mentioned, unfortunately not by myself, but by Ryan. So definitely it's a key important aspect, but you can definitely learn about that. There are so many like courses available, and Ryan as well is a self-taught like computer guy, if you can say so. He's actually a pharmacist as well by training. So keep in mind how skills can be transferred. He's a pharmacist. He actually did study pharmacy at UCL, and then he moved into the automation space. So you can really adapt your expertise and your skill as well. So hope I answered that question. I do have a last. Oh no, there's a couple. Okay, I think there's a one that you can answer perhaps, which is more about the last one. Is
sure.
that
So
except for academic requirements, do I have to do some experimental research during high school?
okay, I'm just going to give it. Well, because these normally I wouldn't go into too much detail about application, but we're getting a lot of questions on this, so I'm happy to answer it. Yeah, so if you apply for a degree through UCAS, you're asked to write a personal statement, and we're the only biochemical engineering department in the UK. So if people are applying to do, obviously you're going to apply for other degrees. They tend to be related ones. So it might be chemical engineering or microbiology or even medicine. So people do ask, what if I'm writing a statement? What should I put in it? What we always advise people to talk about what interests them. So if you're interested in biology, microbiology, if you're interested in health, these kinds of subjects, talk about it. We don't expect people to have experience of biochemical engineering before they join us. We're the only department in the UK that does this. You're not going to get the chance to do the kind of stuff that we do until you come to us. So we totally understand that. But we hope that you have an interest in using biology to solve the world's problems, global health problems, or, the kind of problems that we're looking to solve. We hope that that's the kind of thing that you're interested in studying as well. Biology and chemistry, physics, but also global health and ethics, and these are the kind of things that our students are really, really focused on working on. So, but mainly, I mean, really, it's the academic thing that's really important. But we do like to know that you're interested in these subjects and that they're aligned with us, and hopefully that you know what biochemical engineering is, which you will do after having attended this online lecture. I hope that answers your questions. If you've got any more questions, if there's anything you want to know about admissions or our degrees, please reach out to me directly. We do events throughout the year. We've got an in-person event actually happening on the 27th to 28th of June. You can come along, you can actually see our laboratory and meet the team. It's a fantastic day. It's really good fun. But this is your chance to meet the academics, meet the students. You can do we've got some VR headsets that we're allowing you to do virtual laboratory stuff as well. Should be really good fun. If you can't join in person, we'll do online events as well. I hope that answers your question. Back to you,
Um,
Ivana.
Yeah, we do have just the last one, I think, which we've time to answer, which is as AI technologies evolve, uh, Oh, I lost it. Okay. What would you say are the new challenges in this field and how should researchers adapt to it? It's a very good question. I have to say myself, I'm a pretty new, as I showed you from my career path, from the like new to the kind of like automation space. Well, less than a couple of years, but I definitely think there's a lot going on at the moment and taking place. And I think the challenge is really keeping up, but also with the new technologies and development, you need to do lots of like scoping and like research really to understand what's going on. But I also think the other aspect is that you definitely need technologies and equipment to be able to apply the new technologies to the research. So we definitely are well placed in a sense that we have, in terms of automation, lots of like Tican employed in different areas. So what we're using, my colleague Ryan and I, it's more in upstream, but there's also other Tican that are being used for downstream, which is what I mentioned earlier. So all the purification step. But I think the challenge is really understanding how you can make the most with also the equipment that you have. Of course, those are very like expensive equipment. So we definitely can buy like the newest version, or like, you know, the newest piece of equipment that is out there, but we can do a lot with what is in the department already, which is like lots of equipment, and really being updated and trying to integrate the new technologies in the in what we have at the moment. The Vaxab itself that I mentioned earlier, which is part of like the department, is a really big project with lots of funding. So I'm quite lucky also to have you know, the availability of this funding that really helps a lot with the research as well. So yeah. And I think just probably the last one, I think, Kim, that's for you.
Yeah, so I mean, that's somebody's asking about the minor modules that we have, or the options that we have in our BEng. I think the best thing for this is if you are interested in our degrees, come and join. Make sure you're signed up with UCL, because we will be doing sessions throughout the year when we'll talk about our degrees in detail. So we talk about all the modules that are available on it, careers opportunities, the kind of facilities, the kind of experience you're going to have. So it's not something I can go into detail because not everybody's going to be in the same place as you are, and we really want to use this as a chance to talk about the subject on here. So what I'll do is in a private chat, I'll give you my email address. But if anybody else has got any questions about our degrees, get in touch with me. We do have events where we can go into we can do a deep dive into it and talk about the modules, how they lead on, what what opportunities they offer you, and why people choose to do them. But I think it's best we'll do that in another session. So, and also, I just want to say that people are asking about AI. We've got Matthew Banner. Matty's going to be coming along on the 30th, and he's going to be talking about AI in this sector. I had a quick chat with him last week, and he was talking about how, you know, AI learning models do allow you to do the kind of really huge data mining, which is it's just not possible to do as a human being. When you're dealing with really massive amounts of data, it can be a very, very sophisticated tool with limitations, with opportunities. So he'll go into a lot more detail on that. It's fascinating. He's also he's doing a PhD, but he's also working in the sector as well. So and in fact, I think he's just become a fellow as well. So he's got a really interesting link between what we're doing in academia and what's going on in the sector as well. So he'll give a really interesting insight. And if you want to sign up to those, I'm going to leave Ivana to finish off and have a look at any questions, and I'll go and get a link to sign up to the rest of the talks. But they're on our webpage as well. They're quite easy to find. But Ivana, I'll hand back to you because I'm going to find the page with all the rest of the talks,
Yeah,
just because somebody
yeah,
was asking about that.
yeah. Yeah, exactly. There was a person asking for like signing up to more talks. Yeah, I don't think there are further questions. I think we were able to answer to them all. I do hope you enjoy the talk, and yeah, that you got kind of like an insight of what is currently going on at the Biochemical Engineering Department at UCL. Hope you enjoyed this first session of a series of sessions. I can see that Kim just put the link. So yeah, feel free to sign up for the coming new sessions, and yeah, feel free to get in touch with, of course, Kim, or with myself on LinkedIn, or if you want to hear more about the WhatsApp, you can definitely just go online and look for information with the QR code. And yeah, I hope you do have rest of your day, if that's early morning for you, or otherwise good evening. And yeah, it was a pleasure. Thank you again, Kim, and thank you all.
And a huge thank you to Ivana. This is the first of our series for 2026, and I think we kicked off with a really, and also thank you so much for your questions, your engagement. It makes a huge difference to us, because we know that you're engaging with the subject and we're getting stuff across, but it's really exciting to answer your questions. So thank you for showing up. I hope you have a great week. I hope to see some of the same names appear in some of the webinars later throughout the week. But for now, have a fantastic Monday, have a good week, and I hope to see you soon. Thank you very
Thank
much,
you,
and
everyone.
goodbye.
Bye-bye.